Hello everyone, welcome to Ibdp Biology. We're going to talk about the second part of the intro to cell. Today we're going to discuss about surface area, volume, cell differentiation, and stem cells. So let's get started with the surface area and volume. As you know, cell comes in all shapes and size depending on their species and specialization. But there is actually a limit how large they can grow and their size. As you see on a diagram, the lipids, protein, viruses, bacteria, plant cells, all cells have the certain sizes, and we can see that the certain cells cannot grow beyond their size. That is actually it's a limit for their area, surface area, and volume. Volume is how much space is inside the cell. A cell's volume determines the amount of chemical reactions that can take place in its cytoplasm. These reactions require the input of reactants and the export of waste products. So, thus, the higher the volume, the faster resources are actually consumed. So as you know, the larger number of the reactions, chemical reactions, take place in the cytoplasm. So the reactions that's going on here are called the metabolism of the cell. So the rate of the metabolism really depends on the volume of the cell and cell surface. Surface area, the area outside of the cell called plasma membrane. A cell surface area determines the rate at which molecules can pass through the membrane. If more surface area is available, more reactants and products can diffuse in and out of the environment. Cells with a larger volume requires more precursors for their metabolic reactions. So large volumes require large surface area. Imagine it's like actually balloon. So the inside of the balloon is actually the volume of the cell, what's inside. The outside, which covers the inside substances, is called the surface area. Certain balloons also have their limited size, because we cannot blow up the balloon for like unlimited size. If we just blow so much, the volume will get more, but the surface area cannot take no more volume. That is the case in the cells as well. Their surface area and volume ratio should be proportional to each other. That is the essential part for the cell survival, as you see here. Here, we're going to fill the chart below the cell size, surface area, and volume. So let's see how we see. For example, imagine that the cell has a 1 micrometer. Its surface area is 6 square micrometer, and the volume is the 1 cube micrometer. What will happen to its surface area and volume ratio? Or the others, if we have the 2, 3, 4, 5, 6, 7 micrometer cells, and what about their surface area and volume? Let's see. We're gonna how we are actually calculating the surface area. Remember, the surface area we measure with 6 square s, volume would gonna be cube s. So you are going to measure it like that. So here let's see the size. When the size is only 1 micrometer, the surface area volume ratio is the 6. But as the size is actually growing, the surface area to volume ratio decreases. So surface area volume ratio is small, then the cell is bigger. But if the ratio is big, the cell is small. As you see here, if the size of the cell is small, then the surface area volume ratio is bigger. But if the size of the cell is bigger, so then the surface area volume ratio is little. So here we have the diagram here. So if the let's say if the side length of the size increases, so the surface area volume ratio actually decreases. This is how we can measure this as a graph. To maintain a high surface area volume ratio as they grow, cells have developed a number of adaptations. For example, cells can divide to maintain the same volume but drastically drastically increase the surface area. Or other cells can change their morphology. Cells in the intestines form villi. These are finger-like protrusions that increase surface area. You know, amount of food that we're ingested, so for people that eat so much, their small intestine cannot take too much food. That's why the small intestine adapted a certain thing for more food absorption, this villi. So these villi are just having little microvillus that they are finger-like protrusions, and they increase the surface area and food absorption. Skin cells also flatten out to form the thin films as well. Lung cells in the root of the plants branch out. This gives lung cells access to more air and plant cells more access to water. So you know this flat thing is getting less surface area than the shrinked places. So this is how the example we can give as a surface area and volume ratio. So we have got the review questions that you are going to answer. What role does volume play in cell activity? What about the surface area? Why is maintaining a high surface area volume ratio is important? And what are some ways that cells have adapted to maintain a high surface area volume ratio? Now we're going to look at the differentiations of these cells. You know, individual cells are able to carry out the functions of life, but they can also join to form more complex entities. You might think that, yeah, all of our cells have the same gene, but how it's possible that some of the cells in our body carry out different functions? So this is how we're going to start the differentiation. So here we have the specialized tissue in organism can develop by cell differentiation, which is the process of cells developing the ideal structure needed to carry out a particular function. So in our body, all different cells are coming out from the stem cells. Stem cells are very important because they can differentiate and they can turn into different tissues and cell types, such as nerve cells, red blood cells, muscle fiber cells. There are 220 distinct types of specialized cells in the human body, and they form various types of stem cells spread throughout the tissues. So cell differentiation happens because a different sequence of genes is exposed in different cell types. The control of gene expression is the key to development. Remember this. As I said before, this is the segment of the DNA. So the different segment of DNA carry out different cell types. So when we turn on the certain segment in the DNA, so it switch on and it gives us the epithelial cells. Sometimes we switch on the other side, it gives us the neuron cells. Here, once differentiated cells can further fuse and mature to form specialized tissue. In the example here, cells specialize and fuse to create muscle fibers or liver tissue or organ system. So, stem cells are specialized cells that have the ability to divide and differentiate into different cell types. Zygotes are the most potent cell that we ever have, as they are able to become every cell in our body. So, stem cells have different levels of potency, like totipotent can become any cell type, pluripotent can become most cell types, and multipotent can only differentiate into a class of the cells. So stem cells have great potential for use in medical therapies. For example, stem cells can be differentiated into tissues for implantation, such as nerve cell, cardiac cells, liver cell, blood, muscle, intestinal cells as well. For example, in the stroke, in traumatic brain injury, in baldness, blindness, deafness, majority of these sickness, we can use the stem cells to repair and restore the lost tissues and cells. Embryonic stem cells are made by fertilizing egg with the sperm and allowing the zygote to develop. They have almost unlimited potential and can differentiate into any cell type in the body. So embryonic stem cells, scientists already use the embryonic stem cells, and when they create a zygote, and by zygote they use the certain cell differentiations to restore lost tissues in the body. So almost unlimited differentiation potential, and it has less chance of genetic damage via mutation. But the negative side effects, that more risk of cells becoming a tumor, and removing cells from the embryo kills it. So you need to, if again the cells are losing themselves, you have to go through the same zygote process and embryo process again. Cord blood stem cells are extracted from the umbilical cord of newborn babies, and these cells are pluripotent and can be frozen for storage until needed later in life, as you see, and it can be multipotent, and we can find that we can just make them used for the erythrocytes, for these leukocytes if we need them. Umbilical cord blood cells benefits are easily obtained and stored. Cells come from the patient and so are genetically compatible. Limited quantity in the umbilical cord, but the side effects that storage can be very expensive and limited capacity to differentiate compared to embryonic stem cells, because embryonic stem cells can change into any type of cells, but here in the umbilical cord it says it has a limited capacity. It's actually pluripotent. And adult stem cells are those taken from the adult's own tissue. They are harder to obtain and have less growth potential than the other types. So here, as you see, we can use for the brain, teeth, skin, heart, gut, peripheral blood. So we take it from the bone marrow actually. What happens? Isolate cells from the patients are taken, they are grown in a dish, then reprogramming the factors, waiting for a few weeks. So we take the pluripotent stem cells, we can change them into blood cells, gut cells, and cardiac muscle cells. Benefits are that there is a less chance of tumor development, full compatible with patient's tissue. Removal of cell does not kill the adult, but the side effects are difficult to obtain from different tissues and less growth potential it has. For example, stem cells can be used in Stargardt's disease, which is an inherited condition that results in the degeneration of maculae in the retina. It affects juveniles and can result in blindness if untreated, as you see. There is actually protrusion in the macula, and by this way the sight is actually is lost here. Stargardt disease, as I said, can be treated by stem cells. So in 2010, the US FDA approved stem cell research for treatment of Stargardt disease, and retinal epithelial cells are grown from embryonic stem cells and then implanted. Initial trials have indicated the procedure may be successful, so the next phase of tests have been initiated. By 2020, approximately 200 million people will be affected worldwide. So acute leukemia is a form of cancer that stems from white blood cells inside the bone marrow. Cancerous cells invade the body quickly from the bloodstream. It can be lethal if untreated, and you know that the leukocytes are eating the normal blood erythrocytes, and this causes the acute leukemia. And we can treat this acute leukemia by stem cells as well. Chemotherapy can be used to kill the cancerous cells in the bone marrow. However, this also affects the body's ability to produce white blood cells. So before treatment, stem cells are removed from the marrow and frozen. After treatment, cells are injected back into the body to restore the ability to produce white blood cells. So this is how the leukemia stem cells treatment goes on. We take from the bone marrow the stem cells, then they are multiplied in the cell culture, then transplanted into the patient after chemotherapy. So that becomes like a pluripotent stem cells get into the multipotent immunologic progenitor cells, then they create the erythrocytes. So we have got the review part where you're going to define a stem cell, so discuss the benefits and potential downfalls of the embryonic stem cell, adult stem cell, umbilical cord stem cell. Outline, can stem cells be used to treat Stargardt's disease, and outline, can stem cells be used to treat leukemia. So thanks for watching, guys. Have a nice day. Bye bye.